it's interesting to talk about the future from the Monday still in our northern Alabama. But let me explain first the motivation for our study is we are mostly studying urinary tract infections in those of particular antibiotic resistant type because UTI or urinary tract infections are actually the most common and abundant bacterial infections in the world, with the United States alone registering probably underestimating to 6-7 annual cases a year. That results in over 15% of total antibiotic prescriptions going to treat those usually benign and self-resolving condition. In addition, over 20% of hospital-acquired infections would be also contributing from UTI. And now, globally, and very alarmingly, they're becoming increasingly multidrug-resistant. And up until now, antibiotics so far is the only treatment strategy available, so we need to search for alternatives, and that's our motivation. We know that majority of UTIs are caused by the enteric bacteria coming from GI tract, and main causative uropathogens are gram-negative UPEC or uropathogenic E. coli, Klebsiella pneumonia, and gram-positive Enterococcus faecalis, and while they are coming from the gut microbiome, they're interacting with other well-known microbiomes such as vaginal microbiome, skin microbiome. And for slightly over a decade, we know that the urine is not sterile, and therefore coming to the bladder, they're interacting with urinary tract microbiome. We know that this low biomass microbiome contains a variety of bacteria and other microbes as well. And in women, which are mostly plagued by UTI, this microbiome is dominated by lactobacilli, quite similar to the vaginal microbiome. We now know that very early, starting with the newborn condition, probably several weeks of age, people are colonized in the bladder, and this microbiome appears to change with age and variable among people and associated with different benign and oncogenic urological conditions. And recent studies show that even predisposition to UTIs, and especially recurrency of UTI, might depend on composition of urinary microbiome. And here, we are trying to combine our microbiological knowledge and try to see if this novel microbiome also has colonization resistance, which is characteristic for other more characterized microbiomes, and trying to address the question of whether this microbiome can resist the infection. We were lucky because we were collaborating with several groups, including a group of brilliant Dr. Nas Siddiqui at Duke University, and we collected a repository of urinary bacteria. We don't have a database yet as it is existing for other microbiome, for example, oral microbiome mentioned by Dr. Merritt previously. But we used the Enhanced Cultured Quantitative Technique EQUC to isolate bacteria from catheterized urines from asymptomatic postmenopausal women from 3 cohorts, from those who are healthy and don't have UTIs, and those who have recurrent UTI condition and either take prophylactic antibiotics or not. And we collected over 70 different isolates, which has a very good representative group of the 27 lactobacilli strain, including diverse species, including those abundant in urogenital systems such as L. crispatus, Cassuri, Jensenine, and Inners. And if they sound exotic to you, then you may consider that recently you most likely consumed some of the relative lactobacilli, because those are known to humanity for millennia, and they're used for food fermentation, preparation, preservation, some of them presented this image, and they also now used as a probiotics for gut health, now for vaginal health, and also robustly as a feed additives for livestock. And the reason for these bugs to be so good, because they're really good competitors. They produce lots of noxious components, such as lactic acid. They produce peroxide. They produce toxins such as bacteriocins, surfactants, and so forth. And they inhibit the growth of other microbes, including pathogens, usually. But for the urinary lactobacilli and other bacteria, we don't really have this information. So as microbiologists, we address the question about resistance of bladder microbiome by trying to do initial phenotypic and functional characterization of urinary lactobacilli, and then see how they interact with urinary pathogens. One of the classic assays, which we used, presented here, thanks to the help from BioRender software, and representing this well-diffusion inhibition assay, which literally using the agar plate with the long target bacterium loaded around the plate, and then wells or discs soaked in a drug such as antibiotics or other bacteria, in our case, lactobacilli. And then we can see whether the test component or solution affecting the growth of the target bacteria forming the zone of inhibition, clearance, or killing zone, which we can measure, and therefore can address binary interaction between the pathogen and the commensal lactobacilli strains. One of the real examples, the only one which I'm going to show, is example of E. coli domesticated MG1655 strain interacting with itself or with two lactobacilli strain, showing very thin but continuous confluent loan and inhibition zones, which we then measure for quantitative or qualitative assessments. And now the first results. So we took the lactobacilli, the first which we sequenced after isolation, L. gasseri and L. delbrueckii, and we tested them against a subset of the gram-negative pathogens such as model E. coli, UPEC strains, and Klebsiella, top 52 strain. And what we observed that both L. gasseri and L. delbrueckii inhibit quite robustly the growth of these uropathogens. We then tested gram-positive organism E. faecalis, and then we observed that gasseri inhibit slightly the growth of Enterococcus, while L. delbrueckii does not show a very robust inhibition, if at all. We then were very excited because we found out that L. gasseri came from the healthy cohort of patients, while delbrueckii was isolated from our UTI patient. So we decided that there is a great simplistic hypothesis that strengths of inhibition against model uropathogens can describe how this microbiome affecting uropathogen, and we screen the rest of our urinary lactobacilli in the same conditions. And here I show the summary of testing all of our lactobacilli against the model CFT73 strain, organized by the strains in different species. As you may see, we have different representation from different species, showing that there is a high variability of inhibition strengths for different species. And then we did the same screen repeated for two other major uropathogen, showing the robust inhibition against gram-negative Klebsiella and not as robust and strong inhibition against gram-positive E. faecalis. But then when we overlay this information from screens with the cohort identities from where the strains were isolated, presented here, then we found no correlation of inhibition strengths and health status. And we confirm that by averaging the strengths of inhibition of all the lactobacilli from different cohorts, showing no significant correlation, therefore disproving our simplistic hypothesis that we can in vitro define whether the presence of particular lactobacilli would be resulting in permissiveness to the uropathogens. We now are testing those lactobacilli further, and we are defining the mechanism by which this lactobacilli inhibit growth of the pathogens. And we show that, for example, L. gasseri strains, which I showed before, relies on two different mechanisms of inhibition, such as pH-dependent and cell-dependent inhibition, while the recurrent UTI patient isolate L. delbrueckii, again from earlier, relies mainly on only acidification or lower pH-dependent inhibition. And we're also trying to reproduce this inhibition in more physiologically relevant condition of liquid growth medium for co-culturing of two different bacteria and trying to mimic different microbiome environment by increasing concentration of lactobacilli species, again showing that lactobacilli gasseri and L. delbrueckii would be showing different dynamics in strengths of inhibition in vitro. So we're hoping that that would lead us to explanation of when and how different urinary microbiomes can potentially resist urinary tract infections. But we have to be mindful, if I have a couple minutes left, that this collection was obtained from postmenopausal urines, and we know that postmenopausal women are more susceptible clinically to UTI and recurrency, so we need to do similar kind of experiment with premenopausal microbiomes from healthy and diseased bladders. And we have to realize that the urobiome is complex, and therefore binary interaction may not recapitulate system, especially if we also take into account host-derived and host microbiome interactions, which we'll need to keep in mind to characterize the ecology of this niche. But in summary, The majority of urinary lactobacilli, which we analyzed, inhibit the growth of major uropathogens, especially gram-negative pathogens and less so gram-positive. And this inhibition is working against multidrug-resistant strains, which I didn't show, allowing us to hope that we will be working towards developing a new strategy of treating or preventing UTI by sort of urine probiotics. And we observed tremendous variety of species and even strain level variability of diversity of inhibition of mechanisms and strengths of inhibition. And this kind of information usually does not come easily from amplicon-based sequencing. And therefore, we need more information about composition on strain and species level. And we now need to know and take into account that UTI predisposition should be considered in the context of urinary microbiome and the host environment. With this, let me thank you for listening and thank the people who contributed to this work, including 2 undergraduate in our group who left to continue with higher degrees, Lydia Delani and James Johnson, as well as 2 high school students, Vaishali Osho and Medha Rurajaru, as well as our awesome collaborators, Dr. Nas Siddiqui from Duke University and Dr. Lisa Carson from OHSU, and of course, our funding and you for listening. Thank you.